Calciphylaxis is a rare but potentially fatal disease. It affects patients with chronic kidney disease, especially those on hemodialysis. What is known about calciphylaxis is it is because of a combination of calcification in the skin, also a spontaneous clot formation, and these events lead to development of an ulcer, and that ulcer expands at a fast pace. With the ulcer comes infection and excruciating pain. There is no treatment defined for calciphylaxis, and we don't know how exactly this happens. Our investigation was inspired by a case which I took care at Boston Medical Center. This was a 62-year-old mother who chose hospice care over living with her two daughters because of excruciating pain and the ulcer which kept on increasing. This was a gut-wrenching experience for me, and we started examining how calciphylaxis develops. Along with our collaborators at Mass General Hospital, we examined the sera samples and the skin biopsies of these patients, and then looked in detail the molecular perturbations which happen in these biosamples. With that, we were able to decipher that there are specific signaling alterations in the skin and the sera of these patients, which can give us clue as to how the pathogenesis of calciphylaxis happens.
We took sera from these patients and did proteomic analysis. We found the fascinating upregulation of IL-6. We also confirmed through different techniques like ELISA that the serum levels of IL-6 in all patients who had calciphylaxis were higher than those without calciphylaxis. We wanted to prove the origins of IL-6. Therefore, we took the skin biopsies of patients with calciphylaxis. We then conducted a special transcriptomic receptor ligand analysis. We found a gain in IL-6 signaling in the calciphylaxis skin lesions compared to the controls. We also revealed a potential regulator of IL-6.
We uncovered a novel pathway that starts with IL-6 and induces tissue factor. We validated this finding in the human samples with immunofluorescence assays. We found striking upregulation of IL-6 receptors and tissue factor in calciphylaxis skin biopsies. We wanted to explore IL-6 therapy for reducing tissue factor. We used an approved IL-6 inhibitor called tocilizumab. Treatment with tocilizumab reduced the tissue factor expression. This finding is interesting as tocilizumab can be explored for further therapeutic benefits.
So our investigation gave us more insight into how calciphylaxis develops and what may be the genes and proteins involved in the skin microenvironment driving this process. We found that some of the cytokines, which are specific protein which results in inflammation, is specifically increased in the skin biopsies of calciphylaxis patients. With this, we can develop a panel of biomarkers that can help us to segregate patients who are at higher risk of death or progress of ulcer. And an important advantage of this knowledge gain is to develop targeted therapeutics, which is desperately needed in this field. Overall, I am satisfied that a case inspired us to take this path, which uncovered an interesting observation that can be further developed as a biomarker or a potential therapy for this fatal disease. We really hope that this particular discovery transform this field, which is badly needed for several decades.